Great. Thank you so much, Dr. Morrow. That was a wonderful talk. So as you said, we'll go ahead and be moving on to our Q&A portion of today's webinar. So if you would like to ask a question, please use the Q&A feature, not the chat. That'll just be easier for us to moderate. So let's get started. Our first question. You show all this activity in response to basic sensory stimuli, but have you looked at reward-related activity in the amygdala or something more explicitly tied to emotion?
Yeah, I think that's a question. That's a very common question. In these data, I didn't look at, so the data I collected at the University of Arizona, I wasn't explicitly looking at reward processing because it wasn't the goal of the project. However, now at the Oregon Health and Science University with Dr. Costa, that is one of our explicit goals. We have started to look at not just reward processing on its own, but also how things like reward prediction errors are encoded within amygdala activity. It is very clear that that type of information is encoded in frequency band specific ways, and I'm hoping to sort of expand in the very near future into sort of looking at those more emotion specific processes in these data.
Perfect. Next question. Do you see any coupling between the single unit activity and the LFP oscillations captured in the component time series?
Yeah, so we did start to look at spike field coherence in these data. We took a poster to SFN a couple years ago on that idea. There is some very clear coupling. It seems to be particularly potent at lower frequencies, so delta and theta coupling between spiking activity and the LFP was pretty prominent in these data sets. One thing that we sort of have struggled with is just getting enough neurons simultaneously recorded from the amygdala. It's a quiescent structure. It's hard to get a lot of neurons recorded at one time. But in our upcoming experiments, we're going to be using V probes with 32 channels. We're hoping to get sort of a better collection of single units to try to do some more spike field analyses. It's definitely something that's on our plate.
Great. Next question. Has source separation been used to differentiate between cortical layers?
Yeah, so that's a more common, I think, approach. Obviously, cortex has a nice spatial structure and that there are these layers. I don't know if people have used generalized Eichen decomposition for source separation with cortical layers, but certainly people have used independent components analysis and other source separation techniques to look at layer-specific processing. And the only paper that's coming to mind is the Uzaki paper that used ICA, and they had a lot of success with differentiating activity in the LFP from the different cortical layers. That's a pretty, I think, straightforward and common approach for analyzing activity in cortical structures.
Okay. Next question. How did you determine that 6 millimeters would be enough to span the amygdala? Was the 7T scan necessary for that? And did you experience variability in size between animals?
Yeah, there's definitely variability between animals to a degree, but in general, I've worked with adult rhesus males for most of my career, and for the most part, it's about 6 to 8 millimeters is the dorsoventral expanse as you move through the amygdala. We would always do a structural scan on our animals before we would do any type of recording to make sure that, everything is, intact and healthy before we go in, and that we can use those structural scans to get an estimate of the size of the amygdala. But I would certainly say you need to do those ahead of time. You don't necessarily need to do a 7T scan after the fact to get that sort of readout of the spatial structure of the amygdala.
Okay, follow-up question from that same attendee. Do you have recommendations about which drives to use in combination with the V probes to get clean LFP recordings?
Yeah, so obviously it's going to be dependent on your lab. When I was in graduate school, I used motorized electrode manipulators, or MEMs, from a Thomas recording. They're a company out of Germany. I really liked using those microdrive systems because they gave you a very precise readout on the micron level of where the estimated position of your electrode tip would be. Using pencil drives is with these arrays is great because you can put pencil drives very close to one another and you can potentially get down multiple arrays, but I don't think you necessarily have to go with any one particular drive system. I think, where I am now, at Oregon Health and Science University, we're going to be using a NAND system to deliver these electrodes. But I think you just have to sort of look at your recording setup and try to figure out what's going to work best for you. And I guess I should maybe put in one little plug. Thomas Recording did work with us while I was doing my PhD to create drive systems that allowed for you to basically attach multiple probes to one another on the same drive. So you could put them all at the same dorsiventral depth, which was super useful. And it basically fit into our already existing grid structure. So if I was going to have to make a recommendation, I would go with what I know, which is Thomas Recording, but I don't think that's a requirement for getting these data.
Perfect. Next question. Can you describe more about your methods of implantation? Do you pierce the dura?
So we use cannulae to pierce the dura. We keep our probes contained within, I think it's a 20, I just looked at this. I think there were 23 gauge cannulae. So those were lowered acutely down. Those were used to puncture the dura. We didn't do like a dura slit or anything like that in our recordings. Because I was using a V probe and not an S probe, so the V probes don't have the steel tip, I didn't feel comfortable trying to push those through the dura. I will say there have been times where I've mistakenly not lowered my cannula low enough and tried to push a V probe through dura, and it did not work. They bend, and thankfully you're usually able to catch that before any damage is done. But you have to get those delivered through the dura in some manner, or else you're going to end up breaking your probes.
Perfect. Ariana Andre notes, thank you for a great talk. Could you describe how you go about targeting the amygdala on a daily basis? Also, since you have to go through so much tissue, do you notice damage from repeated penetrations?
Yeah, those are great questions. So with the arrays we have, because they span so much of the dorsal-ventral axis, I generally just try to pick a depth that's going to put my ventral contacts near the base of the basal nucleus. And then I just move around medial, lateral, anterior, posterior to try to not go to the same areas or very similar areas on consecutive days. I think David mentioned in the last talk that you do get tissue fatigue. If you try to go to the same spot, especially the single unit level, you tend to not get as good of isolation on those subsequent days. In the past, what I basically tried to do is just march. I had a recording grid, and I just tried to march through the points in that grid such that I was always staying a couple millimeters away from my previous recording site. However, in my recordings that I'm about to start in the next week or two here in Oregon, we're going to be lowering down. Our initial plan is to put down at least 4 32-channel V probes into the amygdala. And with that, I'm hoping that we basically only have to do a recording or two in order to get enough data to achieve the sort of goals we need. So for that, I'm probably only going to have sort of 2 configurations planned. And if we don't have enough data after that recording, we'll probably let the tissue settle for, I don't know, maybe a week or 2 and then try to go back. But I think the goal with using more of these linear electrode arrays is to not have to go back and record, session after session. It's going to be get in, perform, 3 or 4 total recording sessions with the animals, get enough data, and then be able to seal them up and let them go back to, living their monkey lives.
Great. Next question. With the high cellular density of the amygdala, have you ever tried using tetrode probes for your recordings? And have you ever used chronic recordings in the amygdala?
I have not used tetrodes or chronic recordings. I do know of groups that do. I do think, so with primates, I don't necessarily, my personal opinion is that you don't necessarily need tetrodes. With the 16-channel V probes that we use, 400 micron spacing, I very, very rarely saw waveforms from single units being picked up across multiple channels. I generally didn't have too much problem isolating individual units off of those linear electrode arrays. I mean, occasionally it was a little bit tricky, but I don't think tetrodes are a necessity in the primate amygdala like they are, maybe when you're recording from hippocampus or, maybe a sort of more dense cortical structure. For chronic arrays, again, I've never done that. I know there are plenty of groups that have. The one that pops to mind is David Leopold's group and his the bundle of electrode fibers that he uses, which I think are wonderful. You get great cell stability over time, but the problem with his recording technique is that it can be difficult for you to know exactly the position of each of your electrodes within the amygdala. And for me, that spatial configuration is really important. I really want to know about nucleus anchored processing loops. So if I could have like an array delivered to the amygdala, like a set spatially structured array delivered somehow into the amygdala, where I could be confident in the position of my electrodes, I would definitely go for that. But right now, I don't think that technology is widely available. I know of a group that's trying to get there, but it's certainly not something that's like accessible, at least for me right now.
Okay, great. Maybe you already said this, but why did you use 400 micron spacing?
So the most honest answer here is that 400 micron spacing gave us a solid coverage of the amygdala. So it gives us most of the dorsoventral expanse covered without wasting too many contacts, potentially being outside the amygdala. But there's a lot of, I think, benefits to using that 400 micron spacing. One is you won't pick up the same unit across multiple contacts. So cell sorting is pretty easy. You don't have to worry about duplicating cells across channels. 400 microns means that the LFP that you're picking up across those different channels tends to become fairly unique as you get across a couple of electrodes. So you don't have the exact same signal being picked up on every contact. Now, it's not so much of an issue when you start to apply source separation techniques to your data because you can isolate the sort of independent signals, statistically independent signals from these different networks. But when we started these experiments, we didn't exactly know how to do that. But that's sort of the reason why we went with 400 microns, is just to get good coverage of the amygdala. You could obviously go smaller if you had interest in more local networks. We've even talked a little bit about going larger and trying to have coverage of structures above, dorsal to the amygdala, things like the nucleus basalis and the amygdala at the same time. It's sort of again up to what you want to do in your experiments as to how you design your probe.
Okay, great. I think this is our last question. I think you mentioned you didn't see differences between each area of the amygdala when looking at single unit recordings. Are the neurons in these regions similar in type and function? If not, can you elaborate on why you didn't see this?
Yeah, so the amygdala, I think, it's where I've spent a lot of my time. So I don't know how, I can't necessarily compare it to other brain regions super well, but it is a very, there are a lot of heterogeneous neurons in the amygdala. I think that the reason why we didn't see a whole lot of spatial structure to the signals at the single unit level is just that it's very difficult to get a sampling that's going to be sufficient to sort of get to that level of information. We recorded a couple hundred neurons from a couple different animals, but the primate amygdala has 6 million neurons in it. It's got, as I said, there's 15 compartments in the Paxnos atlas, 15 different nuclei that all have different cytoarchitectural features. Getting enough neurons from each nucleus to be able to look at nucleus-specific processing at the single unit level, I think would just be a very difficult endeavor. Now, if we spent, a lot more time with high-density arrays, I think we could get there, and I certainly think that's worth pursuing. But I think the reason why we didn't see it in those particular data was just that we couldn't get a large enough sample. I think that had we had access to maybe the 64-channel probes, had we been able to put down multiple probes per recording, and had we been able to maybe isolate cells more in the thousands range, maybe we start to see that spatial structure arise. But that's something that's going to have to be addressed, I think, going forward technologically, in order for us to get to those types of questions at the single unit level.
Okay, great. We actually had one more question pop up. I have 16 electrodes in one area, and they show different LFP power. How to do LFP analysis? I mean, I have to do average of all electrodes, or I have to pick up one electrode.
So I'm not sure exactly what the what you're going for there. So I guess there's two things I think might be in that question. One might be a referencing question on whether or not you reference to a single electrode or across all of your electrodes. I use a common average reference, so I average the common signal across all contacts for my data, which is something you can do with the Plexon electrode arrays. In fact, there's different referencing configurations you can put in to either use a reference to the shank of the electrode, or you can set yourself up for common average referencing. You have a little bit of play from Plexon's technology, which is great. But I use a common average reference. When I go to do my analyses, because I'm using this linear electrode array, because these different contacts are picking up some of the same signals, again, I'm going to always go to source separation techniques. For those, I can't necessarily tell you what is right for your data. If you are interested in learning more about them, I would, again, highly recommend checking out Mike Cohen's either YouTube series, or he has a textbook on analyzing neural time series data. And he's also got a linear algebra textbook for source separation. All of those resources, I think, would be great in sort of pointing you in the right direction. But without knowing a lot about your experiments, I couldn't really tell you the best approach for you specifically. Okay,
Great. That was our last question. So thank you so much again, Dr. Morrow, for being here today. That was wonderful. And thank you to everyone else who attended. Please make sure to catch our next installment in our webinar series that will be held next Wednesday at 10 a.m. Central. Dr. Ariana Andre will be giving a talk entitled How Cortical Population Activity is Integrated for Visual Perception and Optogenetic Approach. We'll catch you next time. Thanks, everybody.
Thank you.
Thank you.